Welcome back. We're going to wrap up Chapter 5 on macromolecules with nucleic acids. We might be able to do this in one video. It might actually take two. We'll just have to see how that goes. But we need to talk about nucleic acids. Nucleic acids are the polymer. Specifically, when we talk about nucleic acids, we talk about DNA, deoxyribonucleic acid. Look at that. Nucleic acids right in the name. Or RNA, ribonucleic acids. So, nucleic acids are the polymers. The monomers are nucleotides. So this right here is a nucleotide. Okay, right there. That's a nucleotide. Here's a nucleotide. Here's a nucleotide. Here's a nucleotide. So what I see here is 1, 2, 3, 4 separate nucleotides. Nucleotides are made up of smaller subunits. So remember, nucleotide is the monomer, and that monomer is made up of smaller subunits. So we have a nitrogenous base. So here's our nitrogenous base. We'll talk about which nitrogenous bases exist in DNA and which exist in RNA, but that's a little bit down the road here. We're going to have a 5-carbon or a pentose sugar. In DNA, that'll be deoxyribose. In RNA, it'll just be ribose. The difference there is in oxygen. We'll look at that. And then we're going to have a phosphate group. So when we look at a nucleotide, the sugar and the phosphate, these guys right here, are going to make up the backbone, the sugar-phosphate backbone of the nucleic acid. And then these guys right here, the nitrogenous bases, are going to be what gives each nucleotide its characteristic. Okay, so nucleotide monomers. Remember, the monomer is the nucleotide. We can break that out with another word that's going to be confusing because I've got a nucleic acid and I've got a nucleotide, and now I want you to learn nucleoside. Nucleosides are the nitrogenous base in the sugar. So they're going to be the nitrogenous base in either deoxyribose or ribose. So remember, a nucleotide is a nucleoside plus a phosphate group. When I was first learning that, I had to write it down. I had to draw it out. I had to do it a bunch of different ways because my brain just wanted to flip them around. So however you do that, but do it. But just please do make sure that you've got a handle on nucleic acid versus nucleoside versus nucleotide, and then knowing who makes up a nucleotide. What are the 3 component parts? When we talk about nitrogenous bases, we divide them into 2 families or 2 groupings. We talk about the pyrimidines and we talk about the purines. Pyrimidines have a single ring. Purines have 2 rings. They have a 6-member ring fused to a 5-member ring. The pyrimidines are cytosine, thymine, and uracil. The purines are adenine and guanine. And I think I've said this before, but the sugar in DNA is deoxyribose and the sugar in RNA is ribose. So I need to give you a visual on these nitrogenous bases, these purines and these pyrimidines, and then I'm going to give you a little way to help remember them. On the top, we have the pyrimidines. The pyrimidines have 1 ring. They are cytosine, thymine, and uracil. We'll see cytosine and thymine in DNA. We will see cytosine and uracil in RNA. On the bottom, we have the purines. The 2 purines, nitrogenous bases that we see, are adenine and guanine. Okay. If you look at them, if you just look at what you see right there, you can see that they're different in size, right? So the purines are going to be bigger in size than the pyrimidines. How I learned it to keep them straight was the purines are A and G, right? Adenine and guanine. So all good dogs are pure. Okay? All good dogs are pure. You can remember it however it is you remember it. That's just the tidbit I had years ago, long ago, far away. 2 types of nucleic acids, DNA and RNA. We've said that. Please note that it is not deoxyribose nucleic acid. That's incorrect. There's no ose. You only talk about the ose when you're talking about just the sugar itself. DNA is deoxyribonucleic nucleic acid, RNA is ribonucleic acid. DNA is going to be a recipe. It's going to provide its own directions to replicate itself, and then it's also going to direct the synthesis of RNA. So DNA is going to direct the synthesis of messenger RNA, and through messenger RNA it's going to control protein synthesis. Protein synthesis is going to happen outside of the nucleus in the ribosomes, but that's a later discussion for us. DNA. So we're going to talk about DNA, and we're going to look at it, and then eventually we'll look at RNA as well. Where is DNA? What is DNA? So nuclear DNA is found in the nucleus of most cells. There's another version of DNA, mitochondrial DNA. We'll talk about it when we get to cell structure and organelles, but for right now I want you just to hang on to the fact that most of the DNA is found in the nucleus of the cell. It's going to contain a recipe or coded information formatted in groupings called genes. And those genes, or that coded information, are going to be what programs all of the activity within the cell. DNA is not directly involved in the day-to-day operations of what goes on in a cell. Proteins are responsible for implementing and carrying out those instructions contained within the DNA. So DNA just is rather an oversight piece. It doesn't have a major role day to day. That work is left to the proteins. I think I said this on the last slide, but it's that DNA is going to contain directions for its own replication. So it's going to generate an exact copy of itself, and it's going to pass that exact copy of itself on to the next generation of cells. So we're actually going to have a true cloning of cells. All cells within an organism are going to have the exact same set of instructions. What is going to vary is which of those instructions get turned on or utilized at any given point in time. So I can have 2 houses, I can have 2 refrigerators, 1 in each home. I can have the exact same things in each of those refrigerators, right? But depending upon the needs of the household, you might pull something out of one refrigerator and make one meal, and you might pull something in the other house out of the other refrigerator and make a different meal. So what DNA, which genes get utilized in any given cell depends upon the needs of that cell and the tissues around it. I think this is the spot where I want to take a break. We're at 8 minutes, and then we'll come and we'll just wrap up the last 10 slides or so. I think there's about 10 slides left. So let's do that. We'll pause here and we'll come back and we'll talk about DNA structure as a double helix.